Let's study cellular physiology, and in cellular physiology let's study cell membrane and its components. So cell membrane has three components: lipids, proteins, and carbs. We will just study these components, and after this we can study cell organelles, but that should be like the next video. So let's start with lipids. So the lipids are actually fats, and these are like just fat. And basically the thing is that usually the cell is covered by a bilayer of lipid. Bilayer means two layers. This is first layer, and then there's the second layer, so it means it's two layer. And these two layers are actually asymmetrical in nature. For example, this is one layer. So what can happen is that at one level, at one place, there can be more number of fat cells than other. They are not symmetrical. So lipids, they make up about 40% of the whole cellular membrane, and they are bilayer, and they give membrane the fluidity. And fluidity is really important in cell biology, in cell mobility, because if the cell is not mobile, it won't be able to survive for longer time. If you want your cells to be stable, if you want your cells to be more healthy in nature, they have to be mobile. They have to be typically not rigid. For example, this kind of we want the boundary of the cell to be mobile. We don't want it to be very much immobile. So if we add phosphate group to this lipids, we get phospholipids. That's one type. If we add carbs to the bilayer, we get glycolipids, because glyco is carbs and lipids is lipids. If we add cholesterol, it's just cholesterol. So lipids, they make up about 40% of the whole cell membrane, and usually protein is okay, this is later on. Okay, so phosphate, we add phosphate, it comes as phospholipid. We add carbs, comes as glycolipids. We add cholesterol, it's cholesterol. Let's talk about phospholipids. So there are 5 type of phospholipids I'm going to talk about majorly, and there are many type of phospholipids in nature. It's not just 5 type of lipids, there are many type. The first one I'm going to talk about is lecithin, and I have written that it's very important for the lung maturation, because lecithin plays a big role in manufacturing surfactant. And lecithin is also known as phosphatidylcholine. So choline, phosphatidylcholine is really important in being one of the major major part of surfactant. So if you are less in lecithin, you will not have developed lungs. And if the lecithin is really less, then there will be something called as hyaline membrane disease, which is very common in babies who have who are underdeveloped. So lecithin is if you have less lecithin, your lungs are not going to be developed. Let's talk about another one. It's called sphingomyelin. So as you can see the word sphingomyelin, there is a word called myelin here. So probably I'm talking about the sheath, the myelin sheath here. So again, sphingomyelin, it's used to make nerve cells, again myelin sheath, and they're also found in surfactant as well. But the amount of sphingomyelin found in surfactant is less than lecithin. For example, lecithin can be 80% and sphingomyelin can be like 20%. I'm not saying like this is the ratio. I'm just saying like lecithin is more and sphingo is less. So this is the ratio. Lecithin by sphingo ratio, it should be more than 2, or if it's more than 2 or it's at 2, it's equals to 2, then that means that the fetal lungs are mature. Let's talk about phosphatidyl inositol. It's very common. It's a second messenger of the cell. What is second messenger? Second messenger is just that this is cell, and first message is from the stimulus, Second message from the stimulus, from the boundary of the cell, from the cell membrane to the DNA or whatever, like to the nucleotide or to the nucleus. So that's like second messenger inside the cell. It's very common. Phosphatidylinoside, phosphatidylserine, they are present again. Now they are very important. I remember this thing as siren. So siren are like quite it's, they sing a lot. Siren, if you do know about it, maybe I'm pronouncing it wrong, but siren, and they're present on the inside of bilayer, and if they present on the outside, it means it's a apoptosis signaling. I don't know how to say this, apoptosis or apoptosis, whatever. And we can use annexin 5 staging to look for this kind of staging, to look for this kind of signaling, because if this part of the cell is outside exposed, it means the cell is going to die. And I remember this because serine is like someone who sings, and sings like signaling, and usually it's a signal for death, and usually serines are like horror figures in ocean, something like this. I don't know. Let's talk about cardiolipid. This is very important. Lipid layer, lipid substance. It's most important lipid. It is found in mitochondria of the heart, of the cardiac cells. And why are they important? Because I'm talking about anti-cardiolipin antibodies, and we can actually look for them in this disease called syphilis. And we should do this test called VRDL test for identifying any sort of anti-cardiolipin. So that was phospholipids. Let's talk about a little bit glycolipids. Once you add carbs to the lipids, we get glycolipids. And let's talk about in CNS and GIT. When we talk about in central nervous system, we get cerebro, that is cerebrosides. Now I always remember this as carbs as sides, because it's like we order sides at a restaurant, right? Sides on dishes and like something to eat on side, right? And it's usually something with a lot of carbohydrates in it. So sides is carbs. So cerebrosides, and they are for structural integrity of myelin sheath. So again, I'm talking about here again myelin sheath for myelin sheath. Where was it? The sphingomyelin. Again, myelin sheath, great for myelin sheath. If they are less, they causes Krabbe disease. I don't know how to say this. Maybe I'm wrong. I think I'm wrong pretty much. Krabbe or crab. I don't know. So this causes this disease. Okay. So another one is GIT. They are found in GIT. Again, another glucolipid that is found in GIT is ganglio. Again, sites. And now I just remember this G for GIT and G for ganglio. So these gangliocytes are present as a receptor, and they can be used for cholera toxin. And again, fatal diarrhea can happen. It can kill you. This is GM1 receptor. It's just simple knowledge. That's it. Let's talk about cholesterol. Cholesterol, the one form of cholesterol, that is 7-dehydrocholesterol, are found on the skin, and with the help of sunlight, it transform into vitamin D, and later it goes into kidney, and then blah blah blah. And this is how you get vitamin D, from this cholesterol. If you're less in cholesterol, you're not gonna get anything. So eat your cholesterols. Let's talk about a little bit about this membrane fluidity. Your bilayer should not be rigid, and if you want to make your this lipid layer rigid, you must eat a lot of saturated fat. If you're eating a lot of unsaturated fat, that is like amazing for you. That is fatty acids, omega-3 acids. This is amazing for you because it will increase the mobility. And if you eat a lot of like saturated fat, trans fats, lot of trans fats, lot of saturated fat, it's very bad for you. It decreases the membrane fluidity, and that's it. So what's the work of cholesterol here? Cholesterol is actually neither it doesn't increase and it doesn't decrease. It's actually neutral on the mobility thing. What cholesterol actually does is it tries to maintain the fluidity, you know. It works as a buffer for fluidity. So if you eat a lot of cholesterol, you know, if you eat a lot of good cholesterol, this type of, you know, cholesterol, if you have like good egg yolks and type of cholesterol, you know, it will try to maintain your this you know, this bilayer fluidity, which is really important for you. So try to eat cholesterol, try to eat fatty acids, omega-3, blah blah blah. Okay, let's talk about proteins now. Proteins, they make up about 55%. Again, usually protein is more, you know, 55%. And but where? At myelin. In the myelin, in the nerve cell membrane, lipids make majority of the myelin and protein make 20% of the myelin. So this myelin sheath, you know, of nerve fibers, for example, this sheath is actually made up of lipid. It's not made up of protein. And that's just one, I don't know, it's good point. And by the way, if you like damage your bilayer, cell membrane heal itself and reseals itself. Like lipids, they heal themselves. Okay, protein, they make up about 55% of the whole cell membrane. And protein, there can be three type of protein: trans, on the periphery, on the inside. So trans, periphery, inside, that's it. Trans, periphery, lipid, anchor protein. Okay, inside. Anchored means it's just anchored inside, you know. Okay, let's talk about trans. Not a lot of things to talk. Transmembrane protein, they are, trans means they are throughout the layer, you know, throughout the bilayer, and they work as a lot of ion channels. They work as ion pump, you know, ion come, ion out, like this. Work as hormone receptors, again, because they are outside, you know, so they can like they can identify or maybe look for any hormones outside, you know. They can work as ligand. They work as hormone, they work as ion, they even work as chlorine channels, you know, Cl negative. So they actually can change the electronegativity of the whole cell. So if your chlorine channels are nice, you will have no problems. But if your chlorine channels have, you know, if your Cl minus, the electronegativity of the cell is messed up, you will have this disease called cystic fibrosis. I don't want to talk about a lot of cystic fibrosis, big topic. Okay, so this is transmembrane, you know. And what else? Present mostly in inner mitochondria. Yes, it's present mostly in inner mitochondria, proteins, because mitochondria is one of the very important part. We will talk about mitochondria. So proteins usually present in mitochondria, you know. Of this 55%, mostly present in mitochondria. That's what I'm saying. Let's talk about peripheral protein, this outer one. So this peripheral protein has a lot of functions. First is to support the shape. And which shape? I'm talking about RBC shape, okay? I'm not talking about some shape of some, I don't know, something else. Okay, so we need the peripheral protein and shape of RBC. And if you have bad peripheral proteins, if something is wrong with it, then there's a thing called dystrophin. It's like a structure, and we really need dystrophin. Actually, we need dystrophin for the support of the cell. So if you have problems in this dystrophin, it can cause this disease called Duchenne muscular disease. So I just usually remember Duchenne, D for Duchenne, and D for dystrophin. So if you have problems in dystrophin, it will lead to a disease called DMD, and this is messed up disease because it will eventually like kill you. It will eventually, and it's like it happens in kid a lot. So it's X recessive. Its onset is early, at the age of 5. There is something called Gore sign. It's present. The diaphragm is paralyzed, and because why is it paralyzed? It's paralyzed because there is no support. That's why it gets paralyzed. So eventually the diaphragm is paralyzed. All of your muscles that support your breathing, they can't work, and eventually you die out of breath. So we need peripheral proteins. Another one is that they work as surface receptor. They work as surface receptor because they're on the periphery, they're on the outside, so they can like detect any sort of hormones or any sort of anything. Okay, examples of peripheral proteins: spectrin and ankyrin. They are very too important. They actually are really important for the shape of this RBC and shape of biconcave. I'm talking about shape of RBC. It's biconcave like this. Okay, wait, let me make biconcave. It's a lateral, it's a lateral view. We are looking from the side. So biconcave. Why do we need RBC biconcave? We need this shape because if we don't have the shape, the RBC cannot twist and cannot turn, and they cannot pass through small passages in the body. So we need this kind of shape. So if there is some problem in spectrum and ankyrin, if mutation, then we call it ellipto. Ellipto is like shape, and sphero is also shape. So elliptocytosis. Cytosis is just the cells are becoming, cyto means cells, cis, you understand me. So elliptocytosis and spherocytosis, this can happen. So we don't want that. We want our spectrin and ankyrin to be absolutely all right. Let's talk about the third protein, that is lipid anchored protein, the protein that are inside. Examples. Let's talk about examples first. CD59, CD55, and C8. They need lipid. It means they need someone to anchor on. So they need lipids called phosphatidyl inositol. Do you remember this? Phosphatidyl inositol. See, in second messenger of cell, they are inside. They work as messenger. They are carrying something. What do they carry? They carry some information. They carry this. They don't carry. They just help to anchor these substances. These chemicals. So phosphatidyl inositol for anchoring. They present in hemolysis. Okay, they prevent hemolysis. So wait, let me explain you this better. This is a nice concept. Okay, so this chemical CD59, CD55, C8, they actually they prevent hemolysis. And for the production of these things, we need this gene. It's called PIGA gene. It's a gene. We need this, and this for the production of these, we need this. And if there is some problem in these gene, then there can be hemolysis in the night, and we call this PNH. That is paroxysmal nocturnal hemoglobinuria. And this happens is that why it happens on the night. Okay, it happens the how it happens. Let's talk about this as well. So what happens is that in the night when we sleep, the pH of the blood, it decreases, right? pH of the body decreases. It becomes slightly acidic. And what this acidity does is that it activates the complementary protein system. And this protein system is activated, and then this, especially C5 and C9 protein, they activate something called as RBC membrane complex. And then this attack starts in the night because of low pH, and eventually a hemolysis start, and then that's why we call as paroxysmal nocturnal hemoglobinuria, because hemolysis is happening in the night. So the word paroxysmal here, it means acute. Just remember the word root of paroxysmal is acute or sudden. So the treatment is actually eculizumab and ravulizumab, and eventually the main treatment will be hematopoietic stem cell transplant. This is the hematic, poetic. This is the main treatment. This is just the medicine. So eventually you will need to get this thing done. Okay, carbohydrates. Not going to talk about them a lot. Only 5% present in the whole membrane thing. And if we add carbs to proteins, we get glycoproteins. If we add carbs to lipids, we get glycolipids. Cool. We need glycoprotein for RH system because these are like bigger in, and glycolipids are A, B. Okay, I can be wrong actually at this thing that they can be bigger. I have to confirm that. I have to check that. So RH system works on protein. ABO blood group system works on this lipid. Cool. So I think done.